Hello, this is Marc Schallenberg from the University of Otago, and I'm going to be talking about critical step 1.1.4, understanding how multiple stressors affect ecological resilience and integrity in lakes. This project is jointly carried out by myself, Dave Kelly from the Cawthron Institute, Pete Verberg from NIWA, and David Hamilton from now Griffith University. The partnerships here are really about data sharing, because this project is really a desktop analysis of pre-existing data sets. So the data sets that I'm looking at will come from the Department of Conservation, what we call the cross-departmental research pool data sets for shallow and deep lakes, and also a data set from the Ministry for the Environment, which is their SOE data that they use for snapshots of New Zealand lakes. And NIWA also have some IP input into this SOE data because they do an extensive quality assurance of the SOE data. So yeah, NIWA is also a partner in providing the data used in this study. There's a couple of key linkages which are quite interesting. The resilience aspects of this program also link quite nicely with two national science challenges. The first one, our Land and Water, the Land Use Suitability Program. And that program is really about constraining land use development by looking at the resilience of receiving environments. And so we can see where resilience of receiving environments here links quite nicely to the work done in this program. And also the National Science Challenge, our Biological Heritage Tipping Points program. And that one is about understanding resilience and tipping points leading to regime shifts and loss of biodiversity. So those two programs link quite nicely with the work being done in this project. As I said, it's really about the analysis of pre-existing data sets. So the CDRP data set, so that's split into two groups. One is the shallow and brackish lakes. So these are polymictic lakes. And this information was collected in the first decade of the 2000s and involves about 43 polymictic lakes. And it's around looking at the ecological integrity of these lakes. So it involved one-off samplings of, intensive samplings of the lakes, but for a one-off sampling. So it included looking at water quality, macrophytes, invertebrates, fish, zooplankton, phytoplankton. And so we have a really nice data set across a wide gradient of lakes with very different pressure profiles. The second part of the CDRP data is the deep lakes data, which involved 24 lakes, lakes that seasonally stratify. And again, these data sets were around trying to analyze an ecological integrity in these lakes. So they're also quite a diverse range of variables for these lakes that I can look at. The second data set is the New Zealand Lake State of the Environment monitoring data from the regional councils. And this is, in many cases, more than 20 years worth of temporal data for around 100 lakes. And this lake data set is really going to be looked at in terms of temporal variation to reveal indicators of resilience to eutrophication. So these data are not looked at in much detail in terms of temporal trends. Basically, generally, they're mainly looked at to try and put straight lines through the trends of the data, but there isn't a very sophisticated examination of some of the temporal statistical parameters in the data set, which I think would be quite revealing for understanding resilience. And so that's the idea behind analyzing these temporal. So this is really about understanding multiple stressors and understanding how various factors can confer resilience to eutrophication in our lakes. And one of the ways of looking at this question is illustrated in this analysis of the 43 shallow lakes in the CDRP dataset. And of the lakes that we looked at, we have chlorophyll A on the y-axis and percent pasture in the catchment on the x-axis, which is a kind of general indicator of stress, agricultural, nutrient loading type stress. And what we see here is, I guess, the expected sort of positive relationship between chlorophyll and agricultural development in the catchment. But actually there's quite a lot of variability at the high end of percent pasture. And so the relationship doesn't seem very tight or useful in a predictive sense. And one of the reasons why this occurs is because with the more impacted lakes, lakes are often driven into a behavior where we have regime shifting between alternative stable states. And this is, I think, what's going on in this data set here, because what I've done is I've sized the data points for the different lakes by the macrophyte cover that we measured in each of the lakes. And we can see at the high levels of catchment pressure, there are a number of lakes that actually have quite low chlorophyll readings, and these are the lakes generally with quite high macrophyte biomass. And the ones that have very poor or very high chlorophyll readings are the ones with very low macrophyte biomass. So macrophytes confer a certain amount of resistance or resilience to eutrophication. And we can explore this by building statistical models with macrophytes as a covariate. And here we see that macrophyte by pasture interactions are significant in this relationship, which is exactly what I've just described here. So we have some evidence that macrophytes, for example, confer resilience or resistance to eutrophication, to the pressures of land use change and how they manifest themselves in increasing algal biomass. And in terms of zooplankton, we think that these invasive Daphnia species, the Pulex and Galliata, appear to be quite strong grazers, and so that they may potentially increase resilience to eutrophication by basically taking up or consuming the excess algae produced by increasing nutrient loading. So one of the factors that I'm particularly interested in, and this is also with regard to biomanipulation, one of the other critical steps, is the effect of perch on eutrophication and on water quality. And so again, in these graphs here, we've got percent catchment and pasture as a general indicator of land use stress on lakes, and the total nitrogen, total phosphorus, and chlorophyll A in different lakes. Now this is the CDRP deep lakes data set. And so we do have a positive relationship, as we would expect, between these indicators of trophic status and percent catchment in pasture. But now these bubbles here are scaled to the prevalence of non-native species in these lakes. And we can see that when non-native species are in high prevalence in these lakes, they do appear to have a worse or higher trophic status than the lakes where we don't have such high prevalence of non-native species. So there does seem to be some indication in these data sets here for the deep lakes that non-native species really reduce the resilience and resistance and actually create an enhanced eutrophication in these lakes. Here I've plotted up the same data, but I've plotted up the lakes with perch, which are in red, against the rest of the lakes that don't have perch, again for the same lake data set. And we can see that the lakes with perch tend to have a higher trophic status. Not a lot of data, but this fits nicely with this biomanipulation idea that I discuss in the other critical step on biomanipulation, 1.3.2. So there's some indication here that the presence of perch and the presence of non-native species, fish species in lakes, can have a negative interaction with nutrient loading on lake trophic status. So far we have a number of candidates that are factors that affect resilience and the vulnerability to eutrophication in lakes. And these factors are described in this table here. So things that provide resilience appear to be high macrophyte biomass or cover, short water residence times, which allow greater flushing of nutrients and sediment out of lakes, and low sediment resuspension. So that's probably related to macrophyte biomass, but probably also to lake depth. And so that's in terms of chlorophyll A. In terms of ecological integrity, we have the sediment resuspension, which seems to be related to effects on ecological integrity. In terms of vulnerability to eutrophication, we have low macrophyte biomass, long water residence times, high sediment resuspension, and the presence of perch and goldfish in lakes. In particular, those are the key non-native species that seem to create an enhanced eutrophication response. High sediment resuspension is quite a negative factor for ecological integrity. And when we look at the tendency for lakes to flip between alternate stable states, we have the presence of Egeria densa and the presence of a number of fish species which are highly associated with this flipping. So we have a number of indicator variables here that are key mediators of the nutrient chlorophyll A response in lakes. Now just shifting to the temporal variability, we think that temporal variability has really been an overlooked factor and an overlooked potential analysis that can shed insight on lake resilience. So here we have the 43 shallow lakes in the CDRP data set against the percent pasture in the catchment. And we have not a very strong positive relationship. And one of the reasons why this is the case is that actually when we look at from other data sets, when we look at the temporal variability in chlorophyll A in these lakes, we see that the ones that have high catchment stresses, the ones with high nutrients, in other words, also have really high variability. They have algal blooms, and then they have periods of clear water as well. And this variability, we think, is something that needs to be looked at if we're going to understand the relationship between land use impacts and eutrophication. The lakes that are in red dots here are lakes that are known to have undergone regime shifts. So these are lakes that flip. And so again, flipping is another kind of variability factor that that we really are keen to look at in terms of eutrophication and understanding resilience and variability in lakes. So the way to do this is to actually look at some of the time series in some different lakes. And what you see is that the time series for chlorophyll A, perhaps the best and most direct indicator of eutrophication, are very different in different lakes. The lakes behave extremely differently with regard to how chlorophyll A varies over time. For example, our first example here is Tiwaihora Lake Ellesmere, and this is a lake that is flipped, so it's actually in a very degraded state, as you can see, with the very high chlorophyll levels. Um, but what's going on in this lake is that the chlorophyll just seems to undergo a variation on a number of different time scales. We seem to see this interannual, these interannual changes, which are quite marked. And then we also seem to see these spikes or these peaks of chlorophyll. And I've gone through and I've kind of identified a number of what I'd say are algal blooms within this already very eutrophic system, a number of events where algae kind of rapidly shot up. And so understanding what the factors are that drive these kind of not intermittent but irregular algal blooms, and the factors that drive this interannual variability is something that's of great interest, I think, if we're going to understand how lakes are responding to eutrophication. So yeah, here are some of the key peaks that are really things that need to be looked at in detail, that so far people haven't really spent a lot of time trying to understand these events, these algal blooms. In contrast, we have temporal variability from a lake really close by to Waihora. This is Lake Forsyth, another shallow coastal lake, considered to be in a fairly degraded state. But what we see here immediately is that we have pretty much annual algal blooms occurring in this lake. And really the variability is the extent to which the algal bloom occurs. And so while we have regular algal blooms, what we see is that in some years the blooms are really severe, and in other years they're not very severe. And when you think about it, when you think about factors that actually really matter in terms of management of lakes, it's not so much what the annual average chlorophyll A level would be in a lake like this. It's really about how severe the blooms are. And we can see from this data set is that the severity of the blooms vary quite a lot, although their timing is quite regular, their summer blooms, but their severity varies a lot. And this is reflecting a lake that's flipping back and forth between a macrophyte-dominated stage and an algae-dominated stage. And so this is providing very interesting information in contrast quite starkly to the Te Waihora Lake Ellesmere information that I showed you before. Very different systems behaving in very different ways and showing different information about the resilience of these lakes to change. One more example of a lake that has changed in a rather striking way over time, and this is Lake Waihola south of Dunedin. It's a shallow coastal lake as well. And this lake has a macrophyte community in it. I surveyed it back in 2001, and it had a pretty good, diverse macrophyte community with good coverage of the lake bed. But I've been worried that the macrophyte community might be at risk in this lake. And what I noticed when I went and had a look at the Otago Regional Council's SOE monitoring data for this lake is that the trophic level index in 2010 was extremely high. It went basically an order of magnitude higher into the supertrophic range, and then bounced back down again. And so this is the sort of thing that could indicate that this lake is starting to get into a very vulnerable state to flipping, to potential loss of macrophytes. Has this lake already undergone a summer of severely reduced macrophyte biomass and enhanced algal biomass? It sure looks like it. So again, some interesting temporal information that yields insights as to the resilience of lakes to eutrophication. Finally, the last example is Lake Hayes, which I talked a little bit about in the biomanipulation critical step. But the Secchi disk readings of Lake Hayes, which is considered to be a fairly eutrophic and degraded lake, is showing that in recent times there seems to be a bifurcation in the behavior of this lake. There's been serratium blooms since the mid-2000s, and that has driven water quality way down into very low Secchi depths in the summertime. But interspersed with these very severe algal blooms in summer, we've had summers with extremely clear water, such as 2010 and just the last year, February 2017. So there seems to be this sort of flipping behavior going on as this lake may be entering a recovery phase. It may be recovering but undergoing some kind of major destabilization before the recovery. Again, very interesting temporal trends that are indicative of some major changes and some perhaps loss of resilience in the system. In this case, it might be the loss of a undesirable resilience, a resilience to restoration, which is now being overcome in intermittent years. So we've collected this data, we've obtained permission to use all of this data for our analysis, and the data analysis are ongoing, looking mainly at Secchi depth, TLI, and chlorophyll A in terms of the temporal variability analysis. And we're looking for these signs of rapid regime shifts or flipping behavior, which could indicate low resistance and resilience in lakes. And we're attempting to statistically define these regime shifts in time series, and we'll analyze for drivers of regime shifts and low resistance resilience systems. So once we have these different time series classified into different types of behaviors, we'll try and understand what are the underlying mechanisms that cause them to behave the way they do. And the work is progressing according to plan. There's been a conference presentation in December 2016, and yeah, it seems to be progressing. It's progressing according to plan. We think that there's valuable information in analyzing these large pre-existing data sets, particularly around the issue of resilience or resistance. And we have a question really about how this work could best be of use to stakeholders. And particularly, we're quite curious about what kinds of outputs would be most valuable to the stakeholders with regard to how we could present this work and how we could make this work of greatest value to the stakeholders. Thank you very much.